Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Kallol Gupta. He's an assistant professor of cell biology. He has his own lab, the Kallol Gupta Lab at Yale. And we're going to talk about these deep sea snails that he's studying off the coast of India and, the whole world of biology. So, Kallol, thanks for coming.
Thank you very much for the invite. I'm really excited.
Yeah. So what got you interested in snails and these particular snails? Like, why them?
Yeah, so it was rather an accident that I chanced upon them. Like many PhD students, or at least I should speak for just myself, I had very little idea about what PhD is before I started working. So I remember I was a physical chemist before I started doing my PhD. And then my PhD supervisor said that just because you've done something for 1 year doesn't mean that you have to continue doing it for the rest of your life. And that really inspired me. So, and then when I started joining the lab, there's an idea about looking at natural peptides, because these peptides all bind to, they bind to different membrane proteins and receptors, which is what I'm currently working with. And the idea is these peptides are all neurotoxic. So somewhere or other, they are basically perturbing your cellular physiology. So whatever immobilizes you in a regulated dose, that's basically an anesthetics. So the idea of the motivation behind studying these peptides, and many others actually, which are nociceptive in nature, is to basically understand how these organisms in biology have fine-tuned their arsenal of toxin so that they can actually specifically target different ion channels and receptors in our central nervous system. And in that way, they hijack our physiology. And that sounded like a very interesting question. And it's interesting because my first part of the PhD was basically half of the time I would sail into the deep sea with the fishermen to get the snails, and then get them back in the lab, sacrifice them, get the toxins. And they're basically a very rich concoction, a few hundred polypeptide molecules. And my job was...
Well, quick question here.
Yes.
What do you know of the natural environment of these snails? Like, who tries to prey on them and who are they defending themselves against?
Yeah, it's a very important question. So actually, even in the same natural environment, different kind of snails based on their feeding habit would have different set of toxins. And this is basically a beautiful example of evolution. So for example, a piscivora snail would have a different kind of toxins or different arsenal of toxin than, let's say, a molluscivora snail or a wormivora snail. So based on their feeding habit, it would actually create different set of toxins or a mixture of toxins, which are kind of fine-tuned to that kind of species that they're trying to target. So both the natural habitat as well as the feeding habit is actually very, very critical. One of the key experiment dream experiment that my co-PhD, my late co-PhD supervisor always thought of doing is take one kind of snail which has particular feeding habit and keep it in a controlled environment, a laboratory environment, where you keep feeding. Let's say a molluscivora snail, you start giving it fish. So does it actually change the venom that it's producing within a life cycle or maybe a couple of life cycles? We don't know these things actually that well.
So when does the snail release its venom? Do you know? Is like after a fish tries to ingest it and it poisons its way out? Or
yes,
does the fish taste it and spit it out? Like what happens?
Yeah, so different snails use different kinds of mechanisms. So for example, there's a class of snails which basically inject just the way you would inject a medicine. they would inject, they have a harpoon mechanism using which they would inject these venoms in their prey. Some of them would actually, like a bucket, swallow everything. And once it swallows, that environment is now toxin-filled. So slowly that would sedate their prey. So different snails would use slightly different variation of their preying mechanism.
What do snails eat normally in the deep ocean?
Fish, other snails, worms, whatever you get in the deep sea. And it's fascinating because based on their feeding habit, it would actually change. It's actually really interesting because the first example of these snails in the modern literature comes from a World War diary of an American soldier who was actually posted in, coast of Japan, or in the Pacific coast. And then he encounters these snails, and then he writes in his diaries. And then somehow it's all forgotten. And then somewhere around 1990 and mid-90s, Baltimore Oliveira from University of Utah, who was at that point of time not in Utah, started working with these like a true pharmacologist, a molecular pharmacologist. And he started showing that, look, these snails are basically producing a concoction of molecular libraries, which are now kind of, on an evolutionary time scale, fine-tuned to look at, target different membrane proteins and receptors and hijack our biology.
So what do these snail venoms target? Are they different from, let's say a cobra that would bite you?
Yes.
What kind of venoms are they?
Absolutely. So, yes, they're different. The snake. So I also partly worked with snake venoms during my PhD. The snake venoms, in some way, a bit boring because they all have mostly what they call alpha toxin. They bind to a particular receptor called the nicotinic receptors. It is the receptor which is also responsible for acetylcholine, cholinergic signal transduction. It's a central receptor in our central nervous system and nociception or pain perception. So most of the snake toxins would bind to this receptor. This is also the receptor which gets impaired upon smoking. So it's a big pharmacological target for smoking cessation and related. So what it does, basically, it blocks your pain perception. And this is how I got interested in membrane proteins, because in the first part of my PhD, I was rather an ignorant PhD student whose idea was just to develop mass spectrometric methods to just sequence these proteins, to know their structures, because these venoms that are produced know their structure, what the chemical architecture of these, and knowing that using mass spec and developing newer mass spectrometric methods was my focus. But then what happened halfway into my PhD, the Sri Lankan Civil War broke out. And as a result of that, the Indian fishermen who used to go to the deep sea to collect the fish, they stopped going to the deep sea because that's very close to Sri Lanka and they'll get shelled by the Sri Lankan army. So as a result of that, I stopped getting samples. So I remember I walked up to my PhD supervisor and I asked exactly the same question that you asked, that where are they binding? What are they doing? So then he said that, okay, since you're not getting any sample, why don't you make this question your PhD, the rest of your PhD thesis? And then I started looking. It's not that I discovered where they bind. For many of those, the different kinds, some of the receptors were already known, but some of the novel toxins that I discovered, we found out where they bind. And they typically bind to membrane proteins, which are basically residing at the cellular wall. So, if you compare a cell with a room, just like a room has a wall, the cell has its own wall. And just like a room has a door or a window through which you can communicate with the surroundings, the cell also has its specific doorways and windows, which are mostly made of a specific set of proteins called membrane proteins because they reside at the membrane. Now, 60% of the drugs that are currently available at the market, they all target different kinds of membrane proteins. That's how much important they are to regulate our cellular physiology and different pathophysiological condition. From cancer to different forms of cancer to neurodegeneration, you would see membrane proteins cropping up again and again and again and again, and their impairment cause different physiological deformation. So these toxins are basically binding to these a subset of membrane proteins whose job is basically to propagate neuronal signaling. So if you think about pain perception, which is scientifically called nociception, the idea of nociception is just a neuronal signaling, a particular kind of neural signaling, if you may say, which leads to which are relayed by a specific set of membrane proteins. Now, like sodium channel or potassium channel or nicotinic receptors. And these snails have evolved to generate a large array of molecules, which in like a shotgun manner, it just throws everything at you. Some of them would bind, some of them would not bind. But even if, let's say, 10% of it works in your body, the snail is successful. And it has been successful in blocking the pain perception in your physiology. And at that point of time, you're a helpless prey for the snail.
Wait, why would it mask you from feeling pain? Like if you were envenomated by one of these snails, would you not feel any pain, but slowly you'd lose awareness and go unconscious and then die?
Yes.
What would happen?
Yes, that's exactly what happened. So most of these snails are not deleterious to human beings simply because of the size proportion. So they can't just deliver that much of toxins that we need to sedate a human being. And this is the gray line, right? Anything that is that is making you unconscious in a regulated dose, that's a painkiller. It's exactly the same thing, just the dosage are regulated. And that's
so
exactly
these snails, so if you were able to harness these snails' venom, you could, I guess, have multiple levels. You could maybe do a painkiller with it. Absolutely. You could take it further and do an anesthesia. Absolutely. And you could take it further and then kill someone, which you probably wouldn't want to do. But
yes. So that's precisely why these snails and many other natural peptide libraries are very, very important, because that's exactly what we want to do. And one of the key reasons, the key motivation behind this is exactly what I was mentioning before, that 60% of the drugs currently are targeting membrane proteins. We are, because of the recent, what we call, resolution revolution, where we are determining structures using imaging approaches. Determining structures of proteins have become much easier. Membrane proteins specifically have become much easier than what it used to be, let's say, even 5 years back. So now the next key question that comes, that's standing in front of us is, how do we specifically target one set of membrane proteins without perturbing its very closely related homologs? And that's where these snails have figured it out. They figured it out through a temporal window of evolution, but they can have fine-tuned their genomic library to target only one set of proteins, but not other. I can give you another example of wasp, which I also worked with during my PhD, where they basically they would immobilize their prey not to feed on them, so that they immobilize their prey so that actually they can lay egg inside their prey. And that prey is now just immobilized, but it's still alive. And now that body is basically working as an incubator for that wasp to hatch its egg. And once the eggs are all hatched, then there's no need of the host cell. So the newborns are basically rupturing out of these host organisms, which are immobilized by the venom. So it's a beautiful chemistry or biology, whatever you want to call it, which they have figured out over millions of years of coevolution. And that's really fascinating. And one of the reasons why we're interested in studying both membrane proteins and natural peptide or toxin libraries, because if we want to target proteins specifically, one kind, but without perturbing others, one way to start from scratch, but maybe another way is to look at nature and figure out how other organisms in the nature have already done and learn from them.
So in a way, the snails are kind of like a parasitic wasp. Some of them, they'll infect a creature, stun it, or incapacitate it, but it's still alive, and then they'll lay eggs in it And I guess when the eggs hatch, they eat their way out, or the creature's dead by then, or what happens?
Yes, so the snails don't do that, but there are certain kinds of wasps which would do that. Snails or snakes, their interests are much more immediate. They're hungry, so they want to kill their prey and eat it. Whereas
it's funny. This reminds me. I had a fish tank years ago, and I remember they were fishing it, and all of a sudden snails appeared. I don't know where they came from, and then I saw one day a fish was dead, and there were two snails like suckered onto it. And I thought, how the hell could the snails catch this fish? The snails barely move, and the fish is swimming around. So maybe the same thing happened in my little fish tank years ago that you're talking about.
I would be surprised if you suddenly saw a cone snail appearing in your fish tank. But whatever it is definitely in the same line where they have, co-evolution has basically put a tremendous stress on these organisms to live in extremely hostile environment with their slow speed to develop a unique arsenal which separates them from rest of the food chain. And it's a beautiful chemistry to study.
In thinking about the shape of a snail and how it's so slow compared to a fish, are these proteins expressed on the shell in maybe like a goo? So maybe the fish is mouthing the snail shell, and that's how it gets infected. Or I can't see how the snail would get into a position where its flesh is attached to the fish, or maybe unless the fish starts to swallow it, then maybe it envenomates it. Like what are the different mechanisms that people have found out that snails can envenomate a fish?
Yes. So for the cone snails, they have actually, many of them have developed a harpoon mechanism. So just by throwing an axe, they have venom glands where they synthesize the venom and it's stored in the venom gland. And whenever they want to inject that venom onto their prey, they basically fill that harpoon with the venom and throw it like an axe, and it just stings the prey. And the moment it does that, it's immobilized because it works.
You mean they'll see a fish and they'll go up and spit at it?
Exactly.
And the venom will hit the fish?
Absolutely. That's the majority of that's the mechanism for the majority of the snails. But there are also snails which would, just like a big tub, swallow everything. And then they'll keep it in that environment inside the mouth where there's higher percentage of toxins, which will eventually sedate all the fish and will just swallow all of them. So they're different mechanisms, but the harpoon mechanism is the most common one, which you can compare that with those blow arrows that you get or that you see in some tribes, which basically have those darts, which at the front end are toxins, and they just blow it from a pipe, and it just hits somebody, and immediately the prey is immobilized. It's very, very similar to that.
Yeah, but in the water, how could a harpoon work? I imagine it might be a cloud, like I'm probably wrong, but I would think the snail will go psst, and then like a cloud of venom kind of floats around it, and the fish, swims through it. But you're saying you think underwater there's actual harpoons? Like
yeah, there are videos available. If you're interested, I'll send you some. It's beautiful. Of course, there's more resistance inside water. But they have evolved their muscles and related organs to overcome that resistance to the level that they can survive. So, of course, there's a distance criteria present there. They always wait till the fish comes within the territory within which they can harpoon, and then they...
Well, what I'm surprised about in the water is, I'm picturing a little harpoon, the water changing its path. Like, how could you get a straight path? How could you target anything even a few inches away in the water? I guess maybe it's what does the harpoon itself look like? Is it just composed of a liquid of a higher density, or is it a physical?
No. It's a tissue. It's a tissue-based organ, and inside that you have the venom, which is basically like a needle, which is now injected onto the fish. But yes, you're right.
So it's like a pseudopod or something.
Absolutely. Yes, you're right. So turbulence in the deep, that's why these creatures sometimes would not be in the shallow sea. They would be in the deep sea. where there's probably less perturbance, especially due to human activities, compared to the shallow end of the sea. So they will all you would not get any of the cool snails unless you go deep into the sea, and you have to go quite a depth before you can actually start appearing. So that's why they're very.
In your analysis, then, what do you think will be the most useful thing that will come out of you studying the snails? Will it be a new painkiller, a new anesthetic, or just understanding of how this works, or what?
Yeah. So what I described with the snails is something that I used to do in PhD. Now my research has progressed a bit. What happened when I started understanding how these snails work on membrane proteins? I eventually got interested in membrane proteins. So after I finished my PhD, when I moved to Oxford to do my postdoc, that's when I started getting fascinated by the membrane protein. So it's kind of two sides of the story. In my PhD, I was studying these molecules which perturbs the membrane protein. In my postdoc, I was studying those membrane proteins, which were the target of these molecules and how they assemble in the membrane. And now, over the last 1.5 years, after we have set up our lab, we are very interested in now bringing them together. So one of the key aspects of studying membrane proteins now is to look at how these proteins are interacting with each other in the membrane, how these proteins are assembled in the membrane, what kind of other molecules they're interacting with, and how can we study them directly from the membrane environment. The cellular membrane environment is very, very challenging environment for any biophysicist, simply because there's a lot of lipid, it's not water soluble, and traditionally it has been a major challenge to study anything that is embedded in the membrane. So what we are really interested in doing now is to develop approaches using mass spectrometry, which is in some way a glorified weighing meter, to study these macromolecular protein complexes sitting on the membrane. And then can we use the molecular resolution of mass spectrometers to understand what kind of other molecules binding to these proteins, what happens when these molecules bind, what associates with these proteins, and basically looking at the molecular orchestration of the signaling proteins, which are regulating large number of cellular responses directly from the membrane environment, and how external stimuli are manipulating this signaling cascade, which are taking place in our cell every moment of our life. And one of the key aspects that we are really interested in studying is to look at, to ask the question that, can we take a set of membrane proteins, which are maybe responsible for pain perception, maybe responsible for cellular growth, maybe they're deformed in a particular disease condition, and can we now ask the question that in a particular disease condition, what are the molecules it's interacting with? What happens if I throw another set of possible molecules to that, and then what happens to their cellular, the native cellular organization when I throw these external stimuli inside? And this is where kind of both worlds merges together, because now what we're really interested, we have these set of proteins which are really important for a large number of functional reasons in our physiology. What we're really keen on looking at is that how these molecules are assembled in the membrane. What happens if you throw these large number of toxins present there in biology? How they interact with each other? What happens to their native cellular organization in presence of these 500 or 1,000 toxins present there? A simple challenge here is a snail would produce, let's say, 500 peptides, or let's say 500 toxin molecules. So to be general. So out of these 500, maybe only one or two are binding to your target protein of interest. How do you pinpoint that? How do you pick that one or two out of that 500? So there we are now using the power of molecular power of mass spectrometers so that we can actually detect what kind of molecules are binding to the protein of interest. So that we can now do whatever I did, let's say 10 years back, in a blind search manner, where I would start finding out the sequences and the structures of all the toxin molecule present there and kind of guesstimate based on their structure which one might bind where. But now we are actually going a step ahead. because now we can actually directly look at these proteins and the receptors where these bind. So now we can take these receptors from their cellular environment, throw in all these toxins, and see which one binds, where do they bind, what happens when they bind. And these kind of questions they're very key for two reasons. One, it gives us unique molecular probes to understand cellular physiology. For many of these proteins, we don't really know how their downstream cellular cascades are regulated. That's what these natural toxins give us, a fantastic molecular probe to target exactly one protein of interest, leaving everything aside. Second, we can actually now use this platform as a large-scale drug discovery platform, where we can actually take any natural toxin libraries and throw in these into these membrane concoctions of hundreds and hundreds of proteins, and then specifically ask the question, which one binds to where? And this would give us a fantastic high throughput avenue to now dissect the natural libraries of toxin in a very meaningful manner within a very short period of time. And this is kind of
that's a great idea. Why not get a couple of fish tanks and find out which fish that these snails prey on most often, and have them do it in the tank, and then take the fish before it dies, at various stages, and look at it and see where the toxins are going, what tissues they're preferentially attacking, what cell types. And that would give you a good, scorecard for your analysis and see if it's working.
Yeah, so that's exactly what we're doing. But instead of doing it on a fish, you can do it in a much more targeted. One of the problems of doing it directly on the fish is that you have, on both ends, you're looking at a large number of possible hits. Just in human, we have almost 20% of our genome, almost 25% of our expressed proteins are membrane proteins. So you're looking at 1,000 membrane proteins, and the toxin would be 500. So it's a 1,000 cross 500 problem. Instead, you can say that, look, I know this particular membrane protein is really important for breast cancer. I want to see in this set of toxins, in the snail which produces 500 peptides, is there anything that binds to this protein? If it does, which one binds? What's its structure? Where does it bind? These information would now enable us to directly do a targeted molecular discovery against our protein of interest. Instead of, in a true sense, fishing for your target.
Okay, so, and then what is the end goal?
Yes.
You want to understand how they interface with the membranes.
The two sections in the lab. It's a very new lab. We opened for the last 1.5 years, within which the last 3 months we have been closed. So there are two interests in this. One is fundamental interest in understanding membrane protein biology. And the methods that we are developing and the platforms that we are developing are now enabling us to see things that we could not see before, look at processes that we could not do before. So that is a fundamental need in the field, that can we now look at how these proteins are organized in the membrane, what happens in the presence of different chemical probes, what happens to their organizations and functions and structure function integrity. So that's a fundamental problem which we are very interested in understanding. And a key interest in the lab is to develop techniques and new experimental approaches using which we can address this in a wide number of systems. But then there's another part of the lab which is interested in studying in a much more focused manner, where we have one or two key set of proteins, where we are trying to understand that which kind of molecule would bind, which are the critical drug targets there, where they bind. And there are a large number of proteins where drug targets, we don't even know what's a good topology of a protein to target, or which is a good binding pocket to target when you're developing drugs. So if we understand how nature does it, we can probably then address the question in a completely different way. In a way, we would then mimic the nature instead of doing it from scratch. So that's I would not use the commercial interest. Rather, I would say a practical interest, which we think can lead to a deliverable molecule, or at least a deliverable idea, which can lead to a molecule that can be used in a medical setup to now alleviate different physiological conditions.
Do you have any insight yet on which of these hundreds of peptides seem to be predominantly active, and which are just maybe they're adjuvants to the main ones that are functioning?
So the interesting thing here is everything has a function. And that is the most enigmatic part of this question, that there are 500 molecules. You can think about it. These organisms are under tremendous evolutionary pressure. They have no genomic space to waste. If there is anything that is of no use, they would have filtered it out from their genome by now. The reason for which it's not filtered out is because it serves a purpose. Now, what's really interesting is that we don't really know what are the purposes for many of these polypeptides or top peptide toxins. And that's largely because we didn't have a high throughput way to do a molecular discovery of these toxins and where they bind. And with the experimental methods that we are developing now, we think we are going to be there very soon. We can actually now ask these questions in a much more high throughput manner. And that's what we're interested in understanding, because I don't believe that they're producing something which is of no use to them.
So what are you looking to do high throughput? You're looking at the structure of the proteins? Is that a technology that's yet available, is to determine structure?
Yeah, so the structure determination technologies are now available, and they have been available, especially with cryo-EM, it has become much easier. But any structure determination process is very low throughput. It also requires a large amount of samples. What we are developing in the lab is a novel mass spectrometry approach, which is basically now allowing us to look at a megadalton protein complex, but at the same point of time, we can look at a small drug molecule of few hundred daltons, a few atoms present. So that breadth of molecular resolution that we have is now enabling us to look at these large assemblies of the proteins, but at the same point of time, see which molecules are binding to it. On top of that, we're now developing approaches where we can now take these drug-bound protein complexes and then break it down inside the mass spectrometers, and then find out what exactly is this drug, what is exactly the polypeptide sequence of this drug, where exactly it binds. And these information, once we find it out, that's exactly what is missing right now, because you can take most of these proteins against which the drug binds, and you would look at in PDB, their independent structures are available. But that doesn't really solve the question, because we don't really know, we can't really predict by looking at that structure which of these 500 peptides would bind. And that's where we think the resolution of mass spectrometers are really, really critical, because it gives you unambiguous identification of which are the molecules that are binding to your protein, where are they binding, how many are binding, in fact, if there are any special modifications upon binding. And you can look at all that now from a very physiological environment of the cellular membrane itself. And that's what we are developing in the lab.
But can mass spec, how do you volatilize a giant protein? Or can you selectively cut it and volatilize certain functional groups and then mass spec them? Like, how do you figure this out with a gigantic protein?
Yes. So the kind of mass spectrometry we do, it's slightly different from typical mass spectrometry, which probably many people around the world uses, which is called proteomics. That many people are familiar with. And typically, in a typical proteomic setup, you sacrifice the structure of the protein. So you either denature it by adding organic solvent, or you add a protease which chews up the protein into smaller bits. But what we are doing, it's called native mass spectrometry. So the mass spectrometers, both in the ion optics as well as some of the pressure regimes of the mass spectrometers, are fine-tuned so that we can look at the intact protein complexes going up to as big as the entire ribosome. And that now enables us to look at large protein complexes, keeping their structural integrity. Now, while we are doing this, the mass resolution is also very high. So while we can detect a megadalton protein complex, we can also have a very high mass accuracy at the M over Z axis. So that gives us the confidence of what exactly is binding to that. On top of that, we have now developed approaches in the lab, which is now enabling us to take these protein complexes directly from a cellular membrane-like environment. So you know that they're in the physiological condition. And this has been a major, major challenge in membrane protein field, because the moment you take them out of the membrane, they're not water soluble. So unlike most proteins, they're not stable. We have figured out a way that we can now detect these things directly from the cellular environment itself, or cellular membrane-like environment. On top of that, we have now developed a way where we can now fragment these molecules, these gigantic protein complexes, with the ligand bound, with the molecule bound, directly inside the mass spectrometer. And just like doing a lab sequencing, now we can say which are the molecules binding to it, what is their chemical structure. In fact, we are in the process of developing ways where we can even retain the structural information of which region of the protein where this particular molecular toxin binds. So in that way, you know the structure of the protein, you know the structure of the molecule drug that's binding through mass spec. You would know the number of molecules binding, you would know where they bind. So you really don't need to determine this drug-bound structure of the protein anymore, because you have all the information that's needed to now visualize where exactly the drug binds and how does it bind. That's what we are really, really interested in doing. And this is high throughput, because unlike cryo-EM or crystallographic approaches, the sample requirement is very low, and we really don't need to go through this elaborate preps of making proteins and putting on a grid and related work procedures.
Now that you can identify the various peptides and even ones that are binding, what have you discovered? Anything unusual or interesting about the particular peptides?
Absolutely. One of the key things that is remarkable is that for a very long period of time, we used to think that, so the membranes are all made of lipids, right? So for a very long period of time, we used to think the lipids are just like a brick in the wall. They're just forming a hydrophobic layer which separates aqueous wall. But what's really fascinating to see now is how not just which peptide binds to where, but how that binding is modulated by another layer of regulation of the lipid molecules present there. And that's really fascinating us. We have been working with a couple of transporters which are responsible for nociception. And we can actually now see that there are specific lipids binding to it. There are specific endogenous regulators binding to proteins. And it has been absolutely fascinating. It's early days. Just when we were getting really, really exciting results, we had to shut down everything. We are really hoping to go back to the lab now very, very soon. And it's a fascinating time. Most days, things as it is in the lab, things don't work as we want it to be. But at the end of the day, through that process, we learn something. And we're at this stage now where we are now taking up some of these really challenging proteins to work with, and we're trying to understand that what kind of peptides bind there, or what kind of molecules bind there, what's the role of lipid as a cofactor in modulating these structural assemblies.
What would make a protein hard to work with versus not?
Let me answer that from the perspective of membrane protein. What makes it harder for a membrane protein to work with? And that's because membrane proteins are in a very unique environment. They are residing at a cellular membrane which is completely devoid of water, or mostly devoid of water. So unlike most other proteins, so 75% of a proteome, you can purify them and put it in an aqueous solvent and then do your work with them. For membrane proteins, you can't do that. The moment you strip them off that hydrophobic, greasy lipid and strip them off from that hydrophobic, greasy lipid environment, they would coagulate, they would lose their structure, and they're not functional at that point. So there's no meaningful functional study you can do with them at that point. So the challenge is, how do you keep them out of the, how do you take them out of their native environment, but at the same point of time, keep them native? And people have been working with lots of membrane mimetics to basically mimic the cellular membrane conditions. What we figured out is a way we can now look at things directly from membrane-like environment. And that's where our mass spec setup that we have in the lab is very, very unique, because it now enables us to get rid of this idea of using membrane mimetic, because even on the best days, a mimetic is a mimetic, and we don't really know when it's not doing a good job.
Why can't people do this analysis in an inert oil? Why can't you use like an inert oil or an oil that would solvate, these proteins but not break them apart? Is there something like that exists?
Yeah. So if you want to do it from an inert oil, so there are specific chemical properties of a lipid that that solubilizes the protein. The lipid is both greasy as well as aqueous. So there's a head group so the head of the lipid is polar. It's in contact with the water. Whereas the tail of the lipid is hydrophobic. And that's what is solvating the protein. So the challenge is that creating an environment which produces both kinds of chemical characteristics which are required. And that has been really, really challenging. There has been a huge development in the field over the last 5 to 10 years, where there are newer and smarter detergents that are being made to mimic these lipid environment. But at the end of the day, the moment you take them out of the membrane, they're not the same species, they're not the same chemical environment anymore. And that's the point. We lose many of the critical functions that are available. So, for example, one of the reasons why we are really interested in revisiting our original question, my first scientific question of studying toxic binding to membrane protein, is because if we really want to see how these toxins are targeting a target membrane protein in the physiology, we really need to keep these proteins in that context. Without that, we can't really have a meaningful information. And that precisely is the driving for us to study these things directly from the membrane. Now, the problem of studying directly the membrane is that it just doesn't have your protein of interest. It has, a human cell has close to 1,000 different proteins present in the membrane. How do you now independently study them? And to study them, you need to now take it out of the membrane. So how do you then do that without sacrificing the structure or the function? What we have developed is a way to now gently take them out from their membrane environment inside the mass spectrometer itself, so that it doesn't experience any other perturbing agent for any longer period of time than it must. That's what is now enabling us to now look at these assemblies directly inside mass spectrometer. So our experimental pipeline is now to not just take a protein from the membrane, but throw the membrane itself inside the mass spectrometer, and then break that membrane, use the power of a mass spectrometer as like a hammer, and then hammer down that membrane so that it just disintegrates into pieces, and then we can selectively take a protein of interest based on its mass, and then we can do the downstream studies. That's where the power of the mass spectrometer has been really, really useful for us to develop the protocol that we are developing.
Yeah, that's very cool. Very cool.
Thank you.
Well, Kallol, we're almost out of time. What's the best way for people to find out more about your lab's work?
Yeah, just look at our website or just write to us or write email to me or look at our website. We put everything there. We put things in the bioarchive also before publishing anywhere so that it's open to public and anybody can read it, because that's actually really, really important for propagation of scientific ideas. It comes from the idea of free speech and free knowledge. So it must be available for people to read without the paywall of the scientific journal. So we make sure every work from the lab goes there. And that's the best way to figure it out. And the lab Twitter handle, everything.
Well, very good. Well, Kallol, thanks for coming on the podcast. It was very cool to talk to you. I appreciate it.
Bye-bye. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.